working a lot. So a bit of old story, but he is retired now. So my lab also used to work a lot on it, but I was on the different stream. But I have a pretty good idea about what you're going to talk today. So I'm really interested to listen what you're telling us today.
I hope you like it. Yeah, it's my PhD work. Yeah, I was debating what to present because in my postdoctoral lab, I'm doing more plant epigenetics and CRISPR-based epigenome editing. But then I thought I will just go ahead and do my present my PhD work. Yeah,
that's cool.
So nice meeting you.
Thank you.
I just asked to Pankana. Hello. As the Neha not understand very well Bengali, so we miss our opportunity to talk in the Bengali.
Oh, you can proceed.
This is
I understand Bengali.
In our MSc classes, it also happened. I and Soma and few or others Bengalis are talking the Bengali and they are trying to understand, but now she say that she understand.
Yeah, I think she froze.
Okay.
I think she froze. Okay, it's almost time. It's a couple of minutes left only.
Yes. I'm tested the YouTube and this. Soma is my wife and also a part of team. This Bioingene.
Big journey, amazing platform.
Thank you.
Awesome journey. It's like it's a big one. Yeah.
So I think Neha tried to reconnect, and this time I am able to connect the YouTube, and yes, they are coming on the YouTube. So I'm just allow the attendees to come in the Zoom. Okay, it's coming. So I need 2 minutes more. Soma, have you the introduction? If Neha not able to reconnect, then you go for the introduction. Okay,
probably I will go to YouTube because if you guys are here, so
no problem.
I'm just here to meet you guys.
Thank you.
Okay. So
no problem. We have our speaker now.
Okay, bye guys.
Okay, bye. Okay. Okay. Neha, are you ready? Okay. I just send that one mail and
you started broadcast.
Yes, I just started broadcast, but not. Yes, I started broadcasting, and by 1 minute. Okay, I already sent the mail to all the attendees. Today we have 3,670 application, but obviously actual attendees will lower than that. Let's see how many people that come today. Okay, so I need one more step to share the YouTube in our Facebook page. Then we'll start. Okay, we should start. Very welcome to everyone in Bioingene live webinar series. Today is our 6th webinar in this series. Neha Shoma will assist me to conduct this webinar. I request Neha to introduce our host from University of California.
Thank you, Shiva. Welcome to today's webinar organized by Bioingene. Bioingene is a non-profit organization created to promote plant research worldwide. The webinar series was designed to build a platform from where plant scientists can present their research to the world. We hope future scientists can gain perspective and inspiration by listening to the esteemed plant researchers talk about their scientific accomplishments and their thoughts on the future of plant science. We are grateful that many renowned scientists have accepted our invitation to share their research insights with us. We are also thankful to the audience for the interest in Bioingene webinars. We have received a huge response of over 3,000 registrations for today's webinar. You can attend the webinar through Zoom webinars and live on our YouTube channel. Video recordings of today's event will be available on our YouTube channel. It is our request to all the viewers to please hold all questions for the question and answer round at the end of the presentation. You can type in your questions in the comment box, which will be open right after the presentation. The certificate application link and password will also be available after the presentation. So the topic for today's webinar is Understanding Plant-Virus Interaction to Identify Novel Antiviral Weapons in the Plant Arsenal by Dr. Basudev Ghoshal. Dr. Ghoshal is a plant biologist with a research interest in plant-microbe interaction and plant epigenetics with the underlying goal of applying this knowledge for crop improvement. His active research career began with Professor Swapan Kumar Datta's lab, working on a Rice Biotechnology project in the Department of Botany, University of Calcutta. He got his PhD from University of British Columbia in Canada, where he studied the role of RNA silencing as an antiviral defense response against an important pathogenic plant virus in the lab of Dr. Ellen Sanfason. Following his research interest in translating basic research to applied research for crop improvement, he moved to Professor Steve Jacobson's lab at the University of California, Los Angeles, the USA, for his postdoctoral training. As a postdoc fellow, he is involved in developing CRISPR-based epigenome editing tools and using viruses as delivery agents. Dr. Ghoshal's research work has led to several publications and presentations around the world. Moreover, the tools developed are actively being used for crop improvement. Besides pursuing his research, he mentors several undergraduate students by engaging them in active plant research to help them identify their career goals. Dr. Ghoshal is also actively involved in undergraduate teaching and learning at the University of California, Los Angeles. He serves as a reviewer in several undergraduate research fellowship committees. Now I request Dr. Ghoshal to share his screen and start the talk. Over to you, sir.
Okay, it's visible.
Are you able to?
Yes, yes.
Can you hear me, and are you able to look at my screen? Just a second. I just need to stop the subtitles. Yeah. Is it okay?
Can you increase the size a little bit, the window
okay.
windows?
Now is it okay?
Okay, yes, yes, that's okay, perfect.
Is it okay? And you can hear me, right?
Yes.
Okay, so good morning to everyone in India and whoever is in this, whoever is in the Indian Standard Time Zone. And I believe that there are people who are from other time zones also. So for them, good evening and good afternoon. Before I start my talk, I would like to thank the group of Bioingene for creating such an amazing platform. When Shubha Brother contacted me to present my work, I asked him about the target audience. And he sends me this pie chart. And actually, I was really thrilled to see the pie chart. I felt privileged, but at the same time, it was a little bit nerve-wracking because you can see the diversity in the audience. And then I thought, what should I present to this audience? Should I present my postdoctoral work or should I present my PhD work? And while thinking about it, it was during the time when the lockdown was taking place in US, and I was at home, and I thought probably if I talk about plant-virus interaction, it will be more relevant in this situation. So I selected this topic on plant-virus interaction. So today's talk, what I'm going to talk about is, first I am going to show why is plant-virus interaction important, what is plant-virus interaction. Then I will tell you a short story on plant-virus interaction. This is basically a small part of my research work that I did during my PhD. And finally, I will discuss how can we apply the knowledge of plant-virus interaction. Now, as I noticed that the target audience is diverse, so my first two topics are just very basic backgrounds on plant-virus interaction. So let's start with why is plant-virus interaction important? Now we all know that the human population is increasing rapidly. And with the increasing rate of the increasing population, there is a need for more food. And the question remains is that how can we address this increasing demand of more food? To me, and I think most of you also will think that there might be two strategies to solve this problem. First, by increasing the yield of crops. And we are hearing about different talks in this webinar series about how to increase the yield of crops, and there will be more that will talk about this. The other way is by reduce crop losses. And this is where plant-virus interaction comes into play. Now, why plant-virus interaction? Now, plant viruses are known to cause diseases worldwide to agriculturally important plants. And with the change in the climate, the geographic range is continuously increasing. It is estimated that every year, plant virus diseases cause crop losses up to $30 billion. And still we find that viruses are difficult to control. So we need to make sure, or we need to continue research on plant viruses and their host, and understand the interactions so that we can develop novel disease control strategies. Now, in line with this, just last week, when I was preparing this talk, I saw this particular paper that was published in Molecular Plant. So it's a comment paper. And it talks about, the title of the paper is Pandemics of People and Plant, which is the greater threat to food security. I would encourage you all to read this paper because there are some interesting views about plant-virus interaction. It talks about the current COVID-19 situation, but also it brings about the plant-virus interaction. So kindly read this paper. And you will also find more information about the significance of plant-virus interaction in this paper. Moving forward, let us talk about what is plant-virus interaction. Now, what I have done in this section is to divide it into three subtopics. At first, I will talk about the overview of plant-virus interaction. Then I will talk about one of the important components of plant-virus interaction, that are plant viruses. And I will focus my talk on RNA viruses and their multiplication cycle. Finally, I will talk about the outcomes of plant-virus interaction. So how can we define what is plant-virus interaction? To me, plant-virus interaction is the study of all the mechanisms that underlies the outcomes of these interactions. For example, if a virus infects a plant and the plant develops disease. Now, if you are studying how this disease developed, what are the mechanisms that are taking place, what are the harmful effects, then this is what we study in plant-virus interaction. The other part of plant-virus interaction is also to understand how environment plays an important role in determining these outcomes. So this is mostly, in a simplified term, the plant-virus interaction. Now I will be talking about viruses. Now, what are viruses? Plant viruses are ultra microscopic agents. They multiply only within living host cells, and also for their multiplication, they hijack the plant cellular machineries. The basic components of the plants of the viruses are proteins and nucleic acid, and the viral genome can be either RNA or DNA. So here on the right, you can see electron micrograph of a particular virus called tomato ring spot virus. And these are spherical viruses. And in the bottom, I have a cartoon of the spherical viruses where you can see the viral genome that is encapsulated by a viral code protein covering called capsid. I know all of you remember this, but I thought probably before I get into the details about multiplication cycle of a RNA virus, let us refresh a little bit about the central dogma of molecular biology. So we all know that DNA gets transcribed to RNA, and then RNA gets translated to protein. The virus today I will be talking about, like the multiplication cycle or in the later parts of my talk, is a positive sense single-stranded RNA virus. So what I mean by this is that the viral genome is a RNA. And so from here, the viral RNA can get directly translated into proteins. Let's get a little bit better view of the plant RNA virus multiplication cycle. Now, when a virus infects a plant, the first thing that it does is it releases its viral RNA. This viral RNA is then translated into proteins. Some of these proteins are then used to make multiple copies of the viral RNA. Once the multiple copies are formed, at the same time, some of this viral RNA, in association with some of the proteins called viral code protein, they both assemble to form new virus particles. So this is how basically the virus is multiplying itself. Once it forms these particles in association with other accessory proteins, the virus moves to neighboring cells. And eventually it moves to the different parts of the plant. So this is how within one particular plant, the virus infects first few cells, and then it basically spreads all throughout the plant. The next question then gets raised is that how does the virus infect a new healthy plant? Now viruses need vectors to get transmitted to new healthy plants, and these vectors can be insects, nematodes, or even us human beings. So this is basically a simplified view of the multiplication cycle of RNA virus. But we have to remember a couple of things. So in the previous slide, I showed you how the virus multiplies. But we also have to remember that in a cell, there are other cellular processes taking place. And that is what the virus has to take into consideration. For example, the virus has to develop different strategies to hijack the plant machineries. That is something it must do, because why will the plant suddenly let you hijack its own plant machinery and then let the virus multiply, right? Also, in addition to this, there are plant defense responses that are present within this plant cells, and the virus has to escape or it has to inhibit this plant defense responses. So you can see that the plant virus multiplication cycle actually is much more complicated than I showed you in the previous slide. And the interaction between these different processes actually determines the outcomes of the plant virus interaction. Now, so what are the outcomes of plant virus interaction? To me, broadly, there can be three different outcomes. The first outcome is a case where the plant is severely damaged. Its growth is damaged. It might be damaged to an extent where the plant completely dies. And this is a type of interaction that really worries us. Suppose if you have an economically important plant and that develops a disease where the plant dies, and that's how you get huge crop losses. For example, this is an example. Excuse me. This is an example of a virus infecting cherries. So in the oval, in this red oval, you can see cherries that are not infected by virus. But then you see this other cherries that are infected by virus, and the size of the cherries are affected. And this is affected by a virus called little cherry virus. So you can understand why this virus was called little cherry virus. This is another example where we can see potatoes being infected by potato virus Y. Suppose you go to the market and you see this particular potato. Will you pick this up and bring it to home? And then are you going to eat it? So you can see you will try to avoid this kind of potatoes. Again, you are affecting, again, these viruses are affecting the economy. Another example that I really like is the viral infection caused by cassava mosaic virus on cassava. Now, cassava is currently the third most source of calories in the tropic. And the basically what we are interested in eating from the cassava is the tuber. So you can see here this tuber, usually that's what we human beings eat. But then you can see here in this oval circle, this red circle, that hardly has any, that has a stunted tuber caused by a virus infection. And this again causes huge economic losses, and also this can reduce the yield of the crops to a huge extent. So this is our first kind of outcome where the plants are highly damaged. The second kind of outcome basically is the effect where there is hardly any effect on the plants, where the virus is restricted or it is completely cleared away. This type of outcome can be called resistance. And this mostly takes place in plants where they have particular kind of a specific kind of gene called resistance gene that is specific, that specifically can block the multiplication of a particular kind of virus and restrict its growth within the first few inoculated cells. A classic example of this is the interaction between the tobacco mosaic virus and the N gene. And I would like to tell you all a story because I really like this story. So this was in the year 1930s in New Jersey. What people were finding, well, not people, the farmers were finding is that they were growing tomatoes, and the tomatoes were developing these symptoms. Like here you can see this yellow colored symptoms. Also on the fruit, they were getting this mosaics. This was later found to be caused by a virus called tobacco mosaic virus. And the question was that from where the tobacco mosaic virus is getting infected into these plants. Because in New Jersey, there were no tobacco plants, but they were not cultivating tobacco. Any guesses? If this was in person, I would ask you all. If you have any ideas, you can write it in the chat box too. The answer was this particular cigarette called Double Happiness. What was observed was that the farmers working in the field will go out, they would smoke the cigarettes, and these cigarettes were actually infected by the tobacco mosaic virus. And they would get back into the field and they will work, and unknowingly they will be infecting the tomato plants, and that was causing the disease. This is a picture of a tobacco plant that was that's infected with this particular virus called the tobacco mosaic virus. And let me tell you that tobacco mosaic viruses are the first discovered viruses, and that is how the field of virology, I would say, developed. And why is this? You can see that because tobacco mosaic viruses are very stable. It has been reported that they can be stable in the environment for like 50-60 years. So that is one of the reasons why this virus was one of the viruses that was discovered at the very beginning. But coming back to our story of tobacco, so here you can see that the tobacco mosaic virus is causing great symptoms on this particular tobacco plant. But you won't see the symptoms on all cultivars of tobacco. In certain cases, in certain cultivars, the virus is restricted to a few cells only, like here as shown in this particular lesions. And the reason behind is because these are plants that have a resistance gene called the N gene that basically blocks the virus or restricts the virus within this first few inoculated cells. This other picture is a top view of the same plant, and you can clearly see that all the other leaves are basically symptomless. So the virus is restricted only in the first few inoculated cells. So this example is obviously a great example. Later on, this N gene was introgressed into tomatoes, and there are transgenic tomatoes that express this N gene, and they are resistant to the tobacco mosaic virus. So far, what I have talked about is about the outcomes is that on one extreme, you have a case where the plant survives, the virus is cleared. So you're getting resistance. On the other end, you have the plant dies, the virus multiplies, and this is called susceptible. But there are more, but this is like the two extremes. But more and more examples are emerging where viruses and plants, they are found to be existing in between these two extremes. And this is called tolerance. In tolerance, what is interesting is that the viruses are trying to cause less damage to the plants. And in the same way, plants are helping, or not helping, but the plants are allowing the virus to remain in their cells. And one thing I should say is that somehow it is being noted, it is to be noted is that they are providing advantage to each other. Let me give you an example. This is an example of an interaction between white clover and white clover mosaic virus. Now, white clover mosaic virus shown here in the middle are the white clover mosaic viruses. They infect white clover plants. It was interesting to find that there are these other insects called fungus gnats. Now, fungus gnats are small fruit fly-like insects. They lay their eggs on organic material in the soil, and these eggs in moist conditions hatch, and then the larvae that comes out from these eggs, they start eating the leaves of the plant. So eventually the larvae can eat the whole, all the leaves of the plants, and eventually can kill the plant. So this is really a huge problem. But interestingly, what was observed is that the fungus gnats, the larvae of the fungus gnats, were somehow not preferring plants that were infected with this particular virus. So the virus was helping the plants to get rid, or somehow was creating a condition where the insects didn't like these plants. One of the reasons that was suggested is that these plants, they release certain kind of volatile compounds. And once you have this, in the virus infected plants, what was observed is that in these plants, the volatile compounds that are released, that they are released in a different amount or different profile. And somehow the fungus gnats were detecting this and they were avoiding these plants. So here, the white clover mosaic virus was to some extent providing an advantage to the white clover plant. Another example that we like to think falls in this category is the phenomenon of symptom recovery. So what is symptom recovery? And symptom recovery is one kind of plant virus interaction. In this case, what you find is that the infected cells, they develop symptoms, followed by symptoms on the upper uninoculated or uninfected cells. Eventually, new leaves emerge, and these new leaves are recovered from the infection. And they don't have symptoms. That's why we call them recovered from the infection. They are symptomless. And this phenomenon is called symptom recovery. In this case, we like to add it because we like to add it to the tolerance outcome, because these plants can bear seeds. And I don't think that there are reports showing that there's a difference in the amount of seeds that being produced. So there's less damage on these plants. Also, the other reason why I'm discussing this here is because this is what I worked as a PhD student. So this was my PhD topic, and this is what I studied. And that's what I'm going to talk about in the next few slides. And I'm going to tell you, while studying symptom recovery, how we were able to discover a novel antiviral defense response system in plants. So now we all know what symptom recovery is. This phenomenon of symptom recovery has been observed in several plant virus interactions. It was first reported in the year in the early 20th century. But the mechanism underlying symptom recovery was discovered only in the late 1990s. Dr. David Balcom's lab in Cambridge, they were studying the interaction between tobacco blackring virus and Nicotiana clevelandii plants. They observed that the recovered leaves contained lower levels of viral RNA. So what they found is that the viral RNA is getting cleared in the recovered leaves. They also found out the mechanism that was involved. So what they noticed was that in tobacco blackring virus infected plants, the RNA silencing mechanism, which I'm going to get into the details in the next few slides, cleaves the viral RNA. And finally, the viral RNA is cleared, and that causes recovery. So recovery was attributed to viral RNA clearance by RNA silencing. So let's get into a little bit of details about RNA silencing, or RNAi. So what is RNA silencing? Now, RNA silencing is a molecular mechanism to regulate the expression of genes by small RNAs. This is present in diverse organisms like fungi, plants, animals, and insects. Now, RNA silencing can take place, or it can have its effect, on different parts of gene expression. If it if RNA silencing acts at the transcriptional level, it is called transcriptional gene silencing. If it acts at the post-transcriptional level, it is called post-transcriptional gene silencing. For today's talk, I will focus on post-transcriptional gene silencing, and I will be referring to it as RNA silencing. So let's get a little bit more about the process of post-transcriptional gene silencing, and now I'm going to call it as RNA silencing. So how does this process work? Double-strand RNAs within the cell are recognized by a class of enzymes called dicer-like enzymes. These enzymes cleave the double-strand RNAs to produce smaller RNAs that are 21 to 24 nucleotide long. One strand of this small RNAs then gets loaded into another class of enzymes called Argonaut. The Argonauts, in association with this small RNAs, are called RNA-induced silencing complexes, or RISC. The RISC then targets the mRNA by base pairing. At this step, there can be two effects. First, the RISC can cleave the RNA, that is called, and degrade the RNA, that is called RNA slicing. The other process that can take place is the RISC can block the translation of the RNA, and that is called translation repression. In both cases, the protein synthesis is blocked. And now this is again a simplified look of the RNA silencing mechanism that takes place within plants. Now, this particular RNA silencing process also can act as an antiviral defense response in plants. Now, in this case, the double-stranded RNA precursors that I just talked about in the previous slide are the viral or replication intermediates or secondary structures on viral RNA. So these structures that are formed during the multiplication cycle of the virus or the structures that are present on the RNA are recognized by the RNA silencing pathway, and then they are processed. Now, the role of RNA slicing as an antiviral defense response is well established. It has been observed with many, many viruses. However, the role of RNA silencing, the role of translation repression is not yet known. Now, when I joined my PhD lab, this was the concept. And luckily, while studying symptom recovery, what we discovered or what we found is that translation repression can also act as an antiviral weapon. So basically, this was the first time that we showed that translation repression acting as an antiviral defense response. And that is what I'm going to discuss in the next few slides. So the virus that I worked on was tomato ring spot virus. The tomato ring spot virus is an economically important pathogen. It has a wide host range, and it is economically important because it causes disease on economically important plants in Canada, like raspberries, peaches, etc. Tomato ring spot viruses are positive sense single-stranded RNA virus, and the viral genome is bipartite. Now, when I say bipartite, what I mean is that the viral genome is divided into two RNAs, RNA1 and RNA2. The RNA2 encodes structural proteins like coat protein, or here referred to as the CP. Now, tomato ring spot virus infected Nicotiana benthamiana plants also show this phenomenon of symptom recovery. What you observe is that in the inoculated plants, you do see this kind of symptoms. On the upper uninoculated leaves, after a few days of infection, you get very strong symptom development. And finally, new leaves emerge and they are symptomless. Now, symptom recovery, as I was discussing about the tobacco black ring virus and tobacco black ring virus and the Coachella clevelandii interaction, where also we see symptom recovery. The reason was that you get viral RNA clearance. Now, in my PhD lab, this was before I joined my PhD lab, it was a postdoc called Juan Hogel, who tried to characterize symptom recovery in ToRSV infected plants. So his aim was that to basically see how recovery is taking place in tomato ring spot virus infected plants. And basically he looked at the viral RNA levels and RNA silencing and the RNA silencing mechanism. And he tried to see that whether the similar mechanisms are taking place as observed with the tobacco black ring virus. So the first thing he looked at was viral RNA. So this is a northern blot where you can look at the viral RNA levels. To his surprise, this was really surprising. What he found is that in the recovered leaves, there was no significant reduction of viral RNA. It was not what was shown with tobacco black ring virus. Then he looked at viral derived siRNAs, which are the hallmarks of RNA silencing. So by looking at viral derived siRNAs, you will be able to know that there's active RNA silencing taking place against the viral RNAs. And he did detect viral derived siRNAs, indicating that there is the active RNA silencing mechanism going on. And they published this work but still the question remained is that in tomato ring spot virus infected plants, you see RNA silencing, but there's no viral RNA clearance. So what is causing recovery? So this was the question that I got. And it was a very interesting question. And so I took up this question and I tried to move forward with it. And to address this question, I used a tomato ring spot virus isolate called RASPO1. Now, let me tell you why I took this isolate. Because when plants infected with this isolate when plants were infected with this isolate, they recovered, they showed the symptom recovery phenomenon only at 27 degrees centigrade. So at higher temperatures, plants were recovering. But at lower temperatures, plants succumb to infection and you would get, the plant will be dead. So this is one of the reasons why I selected this, because now I have a situation where I can basically compare between recovered plants and non-recovered plants. The other reason was that it was already known that at higher temperatures, you do get stronger RNA silencing effects than at lower temperatures. So the first experiment I did was I compared the viral RNA and the viral coat protein levels in plants infected by the virus, but kept at these two temperatures. Now so let me discuss the results that I got. First, let me discuss what I got at 21 degrees centigrade. So you see that at 21 degrees centigrade, we didn't get any recovery. We saw severe necrotic symptoms. When we looked at the viral RNA levels, the viral RNA kind of was present in all the samples that we tasted, and it showed a steady increase. A similar pattern was also observed with the coat proteins. Now we detected the coat proteins by using Western blots, and you can see here that there was a steady increase. So severe symptoms at 21 degrees centigrade were associated with steadily increasing levels of viral products. Now let's look at 27 degrees centigrade. What do you see at 27 degrees centigrade? You see a classical symptom recovery phenotype as observed by Huan. As observed by Huan's. So you do get recovery, and then the recovered leaves have no symptoms. We first looked at the viral RNA levels, and you do see viral RNA accumulation in the systemically infected leaves, and then you look at the recovered leaves. There also you do see viral RNA levels, and it was very similar to that we observed in the systemic leaves. So basically, we are not getting any reduction in the viral RNA levels. Then we looked at the viral code protein levels, and this was surprising because in the systemically infected leaves, you do see accumulation of code proteins, but then in the recovered leaves, there's a drastic reduction of code protein accumulation. However, the viral RNA levels were similar in the systemic and the recovered leaves. We also tested for the, we also examined the viral derived siRNAs in these plants, just to check whether there is active RNA silencing mechanism acting towards the virus. At 27 degrees centigrade, at a higher temperature, at 27 degrees, viral derived siRNAs accumulated earlier and at much stronger levels than at 21 degrees centigrade. So to summarize, what we found is symptoms are associated with viral code protein accumulation. Recovery is associated with reduction of viral code protein levels at 27 degrees. Third, recovery is recovery is associated with stronger and earlier accumulation of viral derived siRNAs at a higher temperature. So based on this observation, what I did was, or not, I made a working model and asked two questions. This is just a part of my PhD research work. I will not go into further into the other details that I have done. So this was the first question that we asked. Is reduction of code protein during recovery associated with translation repression? The second question was, is RNA silencing involved in the reduction of the code protein and recovery of TORSV infected plants? So let us focus first on our first question. To do this, we thought we will look into the translational status of viral RNA during various stages of infection. And we thought we will compare the rate of code protein synthesis in this different viral infection stages. Now, it sounded really straightforward, but then we had to think about ways to how we can do this experiment. So what we ended up doing is we ended up doing some in vivo labeling experiment. Now, what we did was we detached the leaves. That were at different stages of virus infection, and we fed them with radioactively labeled methionine for 2 hours. So during these 2 hours, suppose your viral RNA is actively getting translated. So the methionine will get incorporated into this newly synthesized code proteins. However, if your viral RNA is not actively translated, then what will happen is you will get lesser number of code proteins with radioactively labeled methionine. But this sounded straightforward, but here we have another problem, because during these 2 hours, you have other RNAs that are also getting translated, and they are also getting radio labeled. So we took another approach. What we did was we used immunoprecipitation. We used code protein specific antibodies to separate out code proteins from the unbound other cellular proteins. And then we ran them on a SDS page gel. Now, running them on an SDS page gel gives you, excuse me, gives you some gives you some advantage. First of all, when you run them on a SDS page, and then suppose if you stain them with a Kumasi blue stain, what you can get is that will tell you the total amount of coat protein that is present within those leaf tissues that you analyze. So here you can see that there was a drastic reduction in the in the recovered leaves in comparison to the systemically infected leaves. So this basically corresponds to what we saw in our Western blots in a couple of slides before. Now, if you take this particular gel and then you expose it to a film, on the film, you will be able to detect the labeled coat proteins as and they will denote the newly synthesized coat proteins. And this will tell you about the translational status. So here you can see that there was strong labeling that took place in the early stages of infection and not at the later stages of infection. So we were observing translation repression of the viral RNA during late stages of infection. So this obviously was interesting to observe. Then we asked ourselves one more question: is that at 27 degrees centigrade we see this drastic reduction, but at 21 degrees centigrade we don't see the reduction; it's a steady increase. So we asked ourselves that how does the translational status in these particular systemically infected leaves look? So we did the same in vivo labeling experiment in systemically infected plants, in systemically infected leaves. So here you can see that these are systemically infected 6 dpi leaves at different temperatures. So at 21 degrees, and then the other one is at a higher temperature. At 21 degrees centigrade, you do see, when we looked at the total coat protein levels, it was pretty much similar at 21 and 27. When we looked at the radio labeled, when we looked at our autoradiograph film, there was active translation going on at 21 degrees centigrade, but at 27 degrees centigrade, there was repression. So translation repression of viral RNA2 was observed only at 27 degrees centigrade, but not at 21 degrees. So we got back to our working model, and we now have evidence showing that translation repression is taking place, and that is causing the reduction in the viral proteins. This was obviously interesting to find. Then we address our second question, that is RNA silencing involved in the reduction of the coat protein? Now, to address this question, we thought of an experiment where we would basically infect plants that are compromised in the RNA silencing pathway. And then we would look at whether these plants are able to recover. And also we would examine the coat protein levels in these plants. As you can see that the argonaut is one of the main components of the RISC complex, and that's what is required. That is what is required for RNA slicing or translation repression. So we selected argonaut as our target. Now in plants, as you all know, these are huge families of genes. We selected 3 different argonauts: Argonaut 1, Argonaut 2, and Argonaut 4. There was a reason for selecting these 3, because Argonaut 1 was shown to be involved in miRNA and siRNA mediated translation repression of host genes and slicing also. Argonaut 2 was involved in the antiviral defense response. And Argonaut 4, that was involved in the translation repression of another virus, potato virus X, but that was via resistance gene mediated pathway. So we basically selected plants that were lacking each of this gene, or not lacking, I would say, that were downregulated for these genes. because we were doing this experiments in Benthamiana plants, and we had to use this particular approach called virus-induced gene silencing approach to silence this particular genes. So we inoculated the plants with the virus, and then we monitored them for recovery. Interestingly, Argonaut-1 silenced plants did not recover from infection. When we looked at the viral RNA levels from these virus-infected plants or ToRSV-infected plants, we saw similar levels of viral RNA accumulation. But when we looked at the coat protein levels, it was interesting that Argonaut-1 silenced plants only showed the accumulation of coat protein, while none of the other plants that actually recovered from infection had coat protein accumulation in them. So this obviously showed that recovery and the reduction of the coat protein are Argonaut-1 dependent. Next, we asked ourselves that, is it because of translation repression? So we subjected this particular particular leaves to our in vivo labeling experiment. What was interesting was that the labeling, there was stronger labeling in the Argonaut-1 silenced plants in comparison to the control plants, indicating that translation repression of viral RNA2 is dependent on Argonaut-1. So again, when we went back to our working model, and now we have evidence showing that recovery is associated with Argonaut-1 dependent translation repression. And now we proposed a model where we think what is happening is in tomato ringspot virus infected plants, there is Argonaut-1 dependent or RNA silencing is somehow playing a role, and it is causing translation repression of the viral RNA2. And this translation repression is actually causing the reduction in the viral protein concentration. And once you have low levels of viral protein concentration, you are getting recovery. And this was the first example that we were able to show that translation repression of the viral genome can act as an antiviral defense response system in plants. So now we have discovered another weapon in the plant that is being continuously used by the plants. So to summarize all of this data, what we observed is that the phenomenon of symptom recovery is influenced by temperature. It is also dependent on the concentration of the viral proteins. And finally, RNA silencing is involved in the symptom recovery phenomenon, that is for sure. And we did get translation repression by RNA silencing. And we showed that this is an antiviral defense response system. So this is a short story from my PhD work. If you want to get more idea about what else we did, we have published this work, and you can obviously read our paper. Now let's get into our final and last topic, that is, how can we apply the knowledge of plant-virus interaction? Now, obviously, we don't just generate knowledge for the sake of generating knowledge. There must be some use for it. And I think the knowledge that is generated from the plant-virus interaction and studies can be used in mainly 3 different ways. First of all, you can develop or design novel disease control strategies. I think now RNAi is being highly used to control viruses. Also, in plant-virus interaction studies, if we incorporate one more component, that is the vector, because that is what is causing the spread of the disease, right? So if we incorporate that particular component, now we have a way, or we can get some knowledge that will help us to find out ways to restrict the spread of the vector. And that will again help us to design novel disease control strategies. The second thing that we can do is we can take this knowledge and now we can basically make new models that will help us to predict outbreak of diseases. This is, I think this is done for human diseases as well, but now this, we can obviously do it for the plant virus interaction studies. And finally, plant viruses are used as plant viral vectors in biotechnology or in other, in biotechnology a lot. Now, when you are developing a plant viral vector, you just don't use it, or you don't use it based on the information that you have on the plant viruses. You also use the information on the plant part, right? So let me give you some examples about this, about the plant viral vectors. So as we all know, there is a burst or explosion to develop or to use the CRISPR-based systems. In line with the development of the CRISPR-based system, there is a great thirst to use plant viral vectors to deliver this CRISPR-based reagents. This is an active research. Here I am citing just 2 papers, but in the last 4 or 5 months, I saw like 5 or 6 papers that got published using different viruses to deliver CRISPR-based, to deliver CRISPR reagents like Cas9 or the guide RNAs. So this is actively going on. Also, why I wanted to bring this topic is, so you found that I did my PhD on plant virus interaction. I'm still doing a little bit of plant virus interaction studies in addition to plant epigenetics. But what I would like to emphasize is that how I'm using my plant virus interaction expertise in my current lab. So in my current lab, what we are doing is we are developing this CRISPR-based or zinc finger-based targeted epigenome editing tools. We already have published a couple of papers, and I have been a part of these groups where we have developed tools like tools that will target DNA demethylation, that will basically cause targeted DNA demethylation, or we have other tools for targeted DNA methylation. There are more tools that are in the pipeline, and they will soon get published, or in the next 6-7 months, they will get published. So we are developing this tool, and I'm lucky to be a part of this, this group, where I am, on one hand, I am developing these tools, but on the other hand, we also aim to use plant viruses to deliver these reagents for epigenome editing. So that is something I'm actively doing in my current lab. And at this point, I would say that my plant virus interaction knowledge that I had from my PhD is helping me a lot. So you saw that how plant virus interaction can be used. Now, moving on to my my next couple of final slides. So this is a slide that I would like to that I basically got the idea from one of our collaborators, Dr. Rebecca Bott, in the Donald Danforth Center. We are collaborating with them in different projects in US. And her idea, I like this because her idea is that she is using it for plant diseases, but I modified it and tried to use it for plant virus interaction research, where I find that we need to create, we need to have foundational knowledge So which means we need to have basic research undergoing, and then that should be used in the translational research. And this circle should continuously keep going on to move the area of plant-virus interaction research forward. So with this, I will be ending my slide, but before that, I would like to acknowledge the lab where I did this work and my present lab. So I did my PhD in Dr. Ellen Sanderson's lab at the University of British Columbia. It was fun time over there. We used to go for a lot of skiing or snowshoeing, and this is one such event. I really liked it. And then I would like to thank my present PI, Professor Stephen E. Jacobson. Here I am also into several projects where I am developing CRISPR-based epigenome editing tools, as well as I'm developing plant viral delivery tools. This is fun time here. And finally, I would like to thank my undergraduate students. They are, without them, I don't think my research life would be so livable, because they provide energy to me as well. So I'm mentoring a lot of undergraduate students here, and this is mostly the whole list of undergraduate students whom I have mentored, and they have some of them have moved ahead. Some of them are doctors, some of them are researchers now. It's amazing to have them. And with this, finally, I would like to say thank you to all the audience I have here for listening to my talk, and also have the patience to listen to me. And I like this slide at the very end, and this is mostly to the young researchers or young undergraduate students or postgraduate students who are looking at this slide, is that most of the days when we are working in the lab, we ask the question, what is the opposite of eureka? And so most of the times experiments fail. It's like 99% of times. And only 1% of time, you really get a chance to shout out loud and say, Eureka. So thank you. I'm ready to take questions now.
Thank you. Now we have some more questions, like 13 questions. You can choose a few of them from question answer box and can answer them. And I have one question or two questions from YouTube also. So please go for the questions from the question answer box.
So there is a question. I'm not sure what the question is, but this is from Ganesh Satarkar, asking that the difference between RNA and DNA viruses. I would say this is a very basic question. That is, the viral genome is different between the RNA and the DNA viruses. And therefore, the multiplication cycle of the RNA and then the DNA between the RNA virus and the DNA viruses are different. So I'm not going into the details of this question. The potato which, so there is this interesting question that the potato which infected gets infected with potato virus Y can harm humans, can cause harm to humans. This is from Shiva Maurya. I don't think there are any reports of plant viruses infecting humans. So obviously the potato virus Y won't infect the humans. Any known which Gemini virus which we can target using CRISPR? Okay, there is this another question, any known host protein except pCNA which interact with Gemini virus protein, which we can target using CRISPR-Cas technology. At this point, nothing is coming to my mind. So I'm sorry. I think if you Google, if you look at the literature, you might find some. For example, with the RNA viruses, I will go back to RNA viruses because that's where I fear that's what I have worked with a lot, and also currently the projects that I am involved in. One of them actually is with one particular RNA virus. And for RNA viruses, some of the RNA viruses, they do use certain kind of elongation factors like EIF iso4E. They are being targeted. And I think here in the first CRISPR-Cas talk, that question was addressed. The next question we have is from Shiva Maurya. Is symptoms recovery is also possible in susceptible plants? That's actually a very good question. So I am not sure about it, but you know, I had done certain experiments where I would take the plants that were highly infected, that got highly infected at 21 degrees centigrade, and then I will move them to higher temperatures. Sometimes they would recover, but it also depended on the fact that how far the viruses were able to spread. The next question is from Ravindra Sadbhai. Will temperature silence the infection of virus? Now, I don't think that it's temperature that silences the infection of the virus. It's basically the mechanisms, the molecular mechanisms, like whether it's the resistance pathways, like the Argon mediated pathways or RNA silencing mechanism, that gets cleared away. Yeah, that actually clears the viruses. So suppose in this case, in my case, when I was doing the research, at higher temperatures, you would get symptom recovery because you have a higher, stronger RNA silencing taking place at higher temperatures. The next question is, does other environmental factors like photoperiod have any effect on translation repression in case of tomato ring spot virus? Now, we never checked the effect of photoperiods. Never checked the photoperiods with translation repression. But I would say that I was also, while I was in Canada, right after my PhD, I was in a lab for around 9 months in Dr. Deanne Roshan's lab at the University of British Columbia. And one of the projects was actually to study the effect of light on virus infection. And light obviously affects the virus infection, because with light, it's like when you're talking about light, you have to think about chloroplast, and chloroplasts are involved in several several plant defense responses. So you have to keep that in mind, that that there can be effect on the chloroplast, and that can have an effect on the virus infection. This is okay. This is a question from Dr. Akhilesh K. Kusawa. He's a postdoctoral fellow at IISc Bangalore, and his question is, do you think that abiotic stress priming, for example, heat stress priming, will enhance plant resistance or plant virus multiplication or virus movement or code protein synthesis? I would say that this is a good question. There obviously is a possibility, because once you are providing a stress, a particular kind of stress to a plant, then you are actually priming the plant for that particular kind of stress. So you're basically reprogramming the gene expression of that plant. And there can be some genes that are also involved in controlling the other pathways. And so that might have an effect on plant virus multiplication or virus movement. In line with this, I would like to add that there are examples in plants. For example, with Arabidopsis, actually, if you infect Arabidopsis with cucumber mosaic virus, these plants were found to be drought tolerant. So they would sustain drought for a longer period of time. So obviously there can be an effect. viral RNA in presence in recovered leaves, it interferes with any metabolic activities of them. Then we have another question from Isha Madan, that viral RNA in presence in recovered leaves, does it interfere with any metabolic activities of plant? Honestly, we didn't look at other metabolic activities of the plant, but what we were, what our concept was that the virus in the recovered leaves is basically is not getting translated. And we were thinking that some of our other experiments that I didn't talk about here, that the viral protein was actually, if there is viral protein present, then that was inducing certain defense response genes like PR1A. So now if you have low levels of viral proteins, those genes like PR1A were not getting activated. And we obviously have shown this in our work. I just didn't talk about it here. The next question is from Abhishek Samal. Does temperature affect all the virus across plants, or it's only specific for TORSV? No, temperature affects. There are the effect of temperature, I would say, is mostly on all the viruses across plants. Yeah, it's not only TORSV. There are several other examples where temperature was shown to have an effect on the virus multiplication or viral infection. Is there any relation between mutation of plant RNA virus and inoculation of resistant plant virus? I am not sure about this question. And inoculation of resistant plant virus. Mutations of RNA plant virus. I think what is meant here is that since RNA viruses are prone to mutation, is there any relationship between that? And at this point, I'm not sure about the question, so I can obviously try to answer it later if I get the question in a more explicit form. Oh. Is there any mechanism of PR1 expression and signaling involving plant virus? How is that different? Is there any mechanism of PR1A and SA induced signaling involved? How it is different from bacteria? Now, in this case, there is obviously mechanisms for PR1A expression and SA induced signaling in the plant virus interaction. The difference here is there's not, I won't say that there is a huge difference, but obviously you have to think the way a virus infects and the bacteria infects. So a virus doesn't get into the cell, right? So in case of in case of bacteria, the bacteria doesn't get into the cell. A bacteria actually stays outside the plant cell, and then it tries to manipulate the plant cell to get nutrition from the plant cell. So there are receptors on the plant cell that recognize certain receptors, or not receptors, but certain proteins like the flagellins on bacteria, and that triggers like PTI or PAM triggered immunity or effector triggered immunity for the bacteria. But in case of viruses, they are directly injected within the plant cell, and that's a huge difference. So that is obviously one of the major difference, I would say. Please let me know about the interaction between viroid and plants. Okay, so there's one question. Please let me know about the interaction between viroid and plant. Now, this is a great question. And obviously now viroids are basically similar to viruses, but they don't encode for their own protein. And that's, if I started, that's another huge field. Like one of my friend is working on it, and that's his career for the last 15, 18 years. That's what he's working. So if I go into details about all of this, that will be another, that will be a long time to talk about. So I think that's all. I would, before I finish, I would like to, this is from Shilpa N. And I would like to tell her, you will find lots of literature on videoid and plant interaction. So kindly go and look at it. Yeah, I think that's all. Yeah.
I thank you. Thanks for answering from the Zoom box. And I have interesting questions is related to practical also, because most of the questions are very either basic or can be found in elsewhere.
Yeah.
It's totally not related to the topics or your expertise. So there is a question that someone asks that Satabdi Basu in YouTube, that if in the plant ELISA test, the antiserum and conjugate are less, then would you mix the ultra pure or milky water instead of antiserum to check the ELISA and ELISA antiviral test? It is.
So she is asking that whether I should use the millipure or the ultra pure.
Yes, instead of antiserum in ELISA. So I found interesting that
This is interesting, and I never tried it. As a scientist, I wouldn't comment on it. I would say, go and try it and find it for yourself. Because this is a good one.
Yes, and I understand the situation. That's why I put the questions, because sometimes it's happened. Sometimes some of the component is so minutely calculated, supplied by the manufacturer, that some little bit calculation can hamper the, and then a lot of boxes you can't use it. So I don't know about viruses. That's why I put your.
No, it's a good question. I would say that, like, sometimes obviously we do it, but if you're doing it only for once, like only once you're trying it, and that is the first time you are dealing with a virus, and you are doing it for the first time, and you don't know exactly what you're expecting, then that might be tricky. But if you have a positive control, then maybe you can try it. Yeah. I think in this case, if you have a positive control, that will obviously help.
Okay.
Yeah.
So, but thank you. Now we move to our interview session. Are you ready? Okay.
Yes. Because
Soma will conduct that session.
Okay. Yeah.
Hello. It was a very well explained, very illustrative, very nice presentation. I'm sure many of the viewers, and I have also received many positive feedbacks. So we would like to know a little bit about you. Could you kindly say a few words describing your journey so far as a plant scientist?
Okay. So I would start from my school days.
Sure.
So I always loved biology. That was for sure. I don't know why. It was more. I always felt that I can see biology happening, and that's why I always loved biology. And I was studying. So my schooling was in Calcutta, in Kolkata, in Oriental school. And then when I was in grade 12, as you know, I had two choices. One was that everybody goes, I think most people go. I also went for medical school, or the other one was to go for research. And just by looking at me at this point, you know that I didn't qualify my medical entrance exam. So that was out at that point. And then I got into my college, that was Surendranath College, and I went for botany. Now, botany was obviously not my choice. My choice was to study microbiology. But when I got into botany, I realized that botany is actually like it's an umbrella where I was able to study microbiology, virology, genetics, plant biochemistry. So I was getting a lot of lot of knowledge, like I was able to know a lot of things, starting from evolutionary biology. So it encompassed a lot. And also at that time, there were a few professors in my college. I would like to name them Professor Vishwanath Bhomik, Professor Nimai Chandra Barui, and Professor Prabir Kumar Ganguly. They somehow nurtured an interest within me for plant science. And then I moved to, for my master's, I moved to Department of Botany and University of Calcutta, where I started taking, where my special paper was plant physiology, biochemistry, and molecular biology. And that's when I came to know about this scientist. His name was Professor Swapan Kumar Datta. He had just moved to, he was going to move to our department. And I joined his lab. And honestly, that's when I realized that as a plant scientist, I can also contribute to the mankind. He was very energetic, and he actually showed that how you can basically bring plant research straight into, like it was directly applied. And that was very, very fascinating. We would go to different meetings that were basically directly meeting with common people to make them understand the importance of biotechnology, transgenic plants, GMO. So it was amazing. And then for my PhD, I moved to University of British Columbia. And there, what I learned was that how to do science, how to manage time, how to logically think, how to scientifically write. So that's what I learned, and I nurtured it at University of British Columbia. And Ellen, my supervisor, she helped me grow as a scientist. And then after I completed my PhD, I got another option that was a short postdoc that I did in Dr. Deanne Roshan's lab at University of British Columbia. It was a very short one. It was like for 9 months, where I was also studying another virus, cucumber necrosis virus. Excuse me, sorry. And then there also I learned, like, that was a move from one virus to the other that actually broadened my knowledge about viruses, because the virus then I was using was not showing symptom recovery. It was a completely different virus, and it would completely kill the plants. And that was, again, another interesting experience. But by the end of that postdoc, I wanted to learn more about not only plant viruses. My interest was getting more into gene regulation, epigenetics. And that's when I got an offer, and I also took that offer from Professor Steve Jacobson's lab. So right here at this point, I learned a lot of plant epigenetics. And then, so that was like the basic side. So I'm learning plant epigenetics. And at the same time, I am able to develop these tools based on my plant virus interaction studies. I'm developing plant virus delivery tools. And we are also developing CRISPR-based epigenome editing tools. And here in this lab, it's like, I learned so much about gene regulation. I learned about all the cutting edge research, like next gen sequencing, all different kinds of, I would say, cutting edge technology that's going on. That's what I am into at this point, and that's actually helping me to broaden the way I think. And that so far, that's where my research is. I'm enjoying my time in Professor Steve Jacobson's lab, and I will see next where I go. But this has been an amazing, amazing time as a plant scientist, because I have gotten involved with so many good PIs or good mentors, and all of them have somehow contributed in my growth.
That's great. Okay, my next question is evolutionary plants and its pathogens are continuously evolving. According to you, which tools and techniques will help us to better understand plant pathogen interaction?
Um, this is actually again a good question. I would say the 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 new technologies that are coming out, like next gen sequencing, CRISPRs for sure. Um, then I would say metabolomics a little bit. All of this, combining all of this, will help us to get a holistic approach. Because at this point, I don't see research being taking place only in an isolated form. What I mean by this is, if you are doing research and if you're only looking at plant viruses and nothing else, then that's not going to work. Now, at this point, we have to understand or get a holistic approach where we have to combine this tools and techniques like next-gen sequencing, CRISPRs, and again, coming back to metabolomics or the new proteomic techniques that are coming out that are highly sensitive. And that will obviously help us understand the plant pathogen interaction. In addition, I'm not sure how much, but I think somehow single cell sequencing will also help a lot
Yes.
in understanding plant pathogen interaction.
Okay. My next question is that with the knowledge of plant pathogen interaction, is it possible to predict and prevent plant diseases in the future?
Okay. Uh, so first, let me talk about prediction, that whether we are able to predict or not. I would say yes. How? So we have at this point, we have some amount of knowledge on plant virus interaction. We can obviously use this to generate models by computer simulations and to basically predict future diseases. But also I feel like most of the knowledge that we have is not complete, because you see that we take mostly a biased approach. Most of the time we study plant pathogens, like just calling pathogens, because we always think that either microbes are pathogens, but that is not always the case, right? They are also helpful to the plants. Now, we always take this biased approach where we are looking at a particular agricultural crop and studying a pathogen. But there are a great number of pathogens that are present in the wild, like in forest. In forest, you don't get diseased plants. So in a forest, in the wild environment, plants, viruses, or plant or under bacteria, they are all maintaining themselves in an equilibrium. So I think if we can get some idea from those about those pathogens or those microbes, because some of the microbes can jump into our agricultural crops, and that may basically cause havoc. So that's something, if we incorporate that information, and then obviously we will be able to predict. And once we predict, I think we obviously can prevent also. But for that, again, we need to do more research. In line with this, I just thought about, this just came to my mind recently, that computer simulations helps us a lot nowadays. Because I don't remember in which year, but I was attending a conference, and there, one of the scientists for plant viruses, what he was proposing is to make mutations in plant viruses to check the effect. So his idea was that plant viruses are mutating, so let's create new mutations within the viruses and then infect plants and then check whether what is the effect. But I don't think he got funding for that research, because you never know what's going to the virus once you mutate it, and that might get harmful.
Yes. Yeah. Okay, as we are nearing the end of the interview, I have one last question. What would your words of advice be to the young research fellows out there?
Okay. So when you say, I feel like in the world of research, I consider myself also as young, but I believe we are
Sure.
talking about younger,
Yeah.
younger.
Your
Uh,
experiences mainly.
yeah. What I would say is that it's mostly for undergraduate students or postgraduate students or PhDs.
Mm.
There are a few things that I would like to say is that obviously you need to have passion for what you're doing. If you're coming into research, then just keep in mind that this is a 24 hours, 7 days work.
Mm. So true.
Think that don't think that you might get you have to enjoy it. That's my point.
Mm.
Right? You can't complain about your life, because I myself have complained when I was a PhD student. It was like, oh my god. But then at one point I realized the more I complain, the more miserable my life is going to be.
Mm.
Right? So, and then once I stopped uh, complaining that, oh, my life is not moving forward, and that happens to every PhD student. I can tell you, every PhD student so far I have seen in their third or fourth year, they would sometimes say that I'm going to leave my research. It's not going anywhere. So have passion for it. The next thing is make sure that you understand the science, like what is known and then what is not known. Try to get that differentiated early on in your career. Third, I would add is you obviously need to learn techniques like lab work, but at the same time develop other skills like how to make presentations, how to better manage your time, how to I won't all I myself don't like multitasking, but sometimes you have to. So how to multitask while not affecting the quality of your work. So you need to develop those kind of soft skills and just enjoy what you're doing, because as I said in my last slide, I showed that 99% times it is failure. But the one time you're going to get it done, that's like the excitement. And I can tell you that happened to me also. Like I was in my third year of my PhD and my projects were not going anywhere, and then we did this in vivo labeling experiment, and it was like I had no idea how to move forward. And then I discussed with my PI, we came up with this idea, and I tried it. And the first time it worked, what my PI, Dr. Ellen Sanfason, what she said was that, Bashu, enjoy this moment, because you will not get it probably in the next few years. You might not get it. So it came after a long time, but it was really a good feel. So basically, that's what I have to say. And I think if I can do it, everyone who wants to do it can achieve it. It's you have to just have believe in yourself.
That's great. That's so motivating. Thank you. It was simply wonderful having you here. Everybody loved your talk. I can see from the comments. And I hope we will see you again in the future on Bioingene.
For sure. I would. I didn't add it to my talk, but while preparing the talk, I was thinking that there will be more. I'm at a point where it's like I'm not able to discuss about my unpublished data, so I didn't present it also. And probably in the next few months, I will have more stuff coming out, and I will obviously would like to present my data again in front of this diverse community. And before leaving, I would like to thank the Bioingene group again. It's again an amazing platform. And to all of you, I must say that this is actually, if I remember right, I moved to Canada in the year 2009, but somehow every time I went back to India, there was either some emergency or I didn't get time. I never presented my work back in India. So this is the first time I am presenting my work to Indian audience. So it's amazing. So thank you very much.
Okay, let's go back to Shubha now.
Okay.
Today we have so many audience. It's houseful in Zoom platform, and so many audience. I 400 and live participants. As I remembered. So we also start to receiving the certificate and feedback applications. I will let you know that how much active viewers are listen to your talk. And these all are available very shortly within 2 days. I think within 2 days in our YouTube, and they can view it more and more by the plant researcher, and those are the interest in plant research. So I am very thankful to you that you believe on us and you give your talk in this platform. And when it is build that platform, then easy to approach anybody else. But I am very thankful to those all the 15 or 20 speakers in our queue that are in August, in September, and I'm very thankful to all of you that believe in Bioingene that we can do that. And I'm happy that we shut out some processes like certificate distribution, how to tally the actual viewers, and this it is also learning for us also. So we are now in the stage that we can invite others, and obviously I'm going to invite you all once again in future.
Okay.
Please let me know when you're comfortable to disclose your work, so it will be a part 2. And it very interesting when someone listen your part 1 and listen your part 2. It also motivate them also that okay, I'm doing this and now I'm doing that. So it's a where the science goes, where the flow are moving to the scientific world. It will definitely it will help, because in PhD we're for 4 to 5 years. So this also help to understand. It's my experience when I got the opportunity to go to the ED, then I explored my disability. Okay, I can interact to others, I can approach to others, and I can build a platform where we all together, because it's for betterment of plant science research. And as you know, it is not for profit. We are just, everything is here free. So I'm very happy you accept and deliver your wonderful talk. It will definitely help us and all the viewers.
I just want to add 2 things before we end up. One obviously is I missed it when Shoma was asking me questions. Is I would like to. This is just an open suggestion to all my young research fellows, is that keep an open mindset, and as Shubhabrata said, that just remember that every mistake you do, you are learning something new. That's what helps me a lot. And before I just keep adding things. And finally, if I would like to help again undergrad students and postgraduate students, or if anyone has any question, they can obviously contact me. I am available through LinkedIn. If they want, then go ahead and you can obviously give my details to them. Yeah.
In YouTube, I got a message that someone tried to collaborate with you. So I tell her that you can go for it. The availability of your details is available on the website, so you can go for it. Because I don't ask to you to whether I am sharing your information or not. So I just surfacely tell him that you can go and you can collaborate.
No, so, I'm open to that.
Yes. Thank you for being with us. And today we I want to end that meeting, and very soon I think we will meet again here. And we have some 2-3 points to discuss with the audience, so you can lead that meeting and I'm sure.
Thank you very much. Bye, Shoma, Neha, and
Thank
Shubhabrato.
you.
Nice meeting you all. Bye-bye. And thanks
Bye.
to the audience.
Now I'm request to Shoma if you give some instruction, whether it should be short because everybody now knows that how they can get the certificate on anything. So it should be short that you can try or you can add.
Mute. All right. So you know that we have changed the certificate application, certificate sending process. Previously, we were sending it by mail, but now you can download your certificate. So, for example, for today's webinar, we will receive all the certificate applications and the feedbacks, which has been provided to you, the password and everything. So you have the password, you have the link. Please apply for the certificate if you want to. And we will receive the form, your form, and we will make the certificates and we will upload it in our Google Drive, which the link for which we will share it in our website. The second thing which I want to clarify is that if you have not registered before the webinar, then I'm sorry, you won't be applicable or eligible to receive the certificate, even if you apply for it. If only the previously registered attendees for any particular webinar, only the previously registered attendees will be entitled to receive the certificate. That is the procedure. It's very simple and everything is free. So we expect you to understand this. All the registrations are ongoing. Please visit our website. The registrations are ongoing. You can register. The next webinar is on 15th. Today is 13th. Next webinar is on 15th. Same time, 11 a.m. The registration for that is also open. Please go to our website and register. Also, new webinars are upcoming for the month of August. We have opened up the registration for the month of August. You can also find the new webinars. All the information has been provided in the website. Please visit our website. Everything is there on the home page. Please register for the August webinars also. And today's webinar will be shortly uploaded on YouTube, on our YouTube channel. So if you have subscribed, then you will definitely receive the alert whenever a new video is uploaded. I would recommend if you have not subscribed, then please subscribe to our channel so that you don't miss out on any videos being uploaded. You can share and you can comment, you can share. We would love to hear from you, and please register for the webinar, for the next webinars if you want to apply for the certificates or you want to be eligible for certificate application. So I think that's it. So it's 12:45 p.m. in India now, and the certificate application link or the feedback form is open now. You can apply, but it will be only open till 2 p.m. Indian time, 2 p.m. Almost for more than an hour 15 minutes. So please hurry if you want to apply. It will be closed in 1 hour and 15 minutes time.
See you next week.
And I'm looking forward to having you guys over for the next webinar that is on 15th. I'm really excited for the next webinar. See you then. And bye for now. Thanks. Okay.
Thank you, viewers. now I'm ending this webinar. We see you all in 15th, our next webinar. Registration is ongoing. Go and register, because register attendees only get the certificate if you want to get the certificate. Otherwise, our webinars are all free for everybody. You can view it from Zoom and YouTube from our channel. Bye-bye.